we're 1 minute away from the time where we promised in the schedule that you would be speaking. So I'll leave it to you, Taylor.
All right. Okay, so we can get started, boss. Sorry, I'm just trying to do too many things at once. Okay, so I think we're about ready to get started. So today I'm going to talk about the CMAX project, which was a project that came out of GLEON that really focused on deep chlorophyll maxima. And then if I have time at the end, I'm also going to touch on a couple things that I personally learned about doing GLEON science from being involved in this project. So for those of you who don't know me, my name is Taylor Leach. I'm a postdoc at Rensselaer Polytechnic Institute, which is in New York. So in a lot of lakes and in many parts of the ocean, vertical profiles in chlorophyll typically often show these really pronounced peaks deep in the water column that we call deep chlorophyll maximum or DCMs. They have a variety of names, but I'm going to call them DCMs because that's what's stuck in my head at this point. And DCMs are ecologically important for a number of different reasons. They form important vertical habitat gradients for consumers, such as zooplankton. They themselves can be really important sites of primary production. Some studies have estimated in systems that DCMs can make upwards of 60% of the aerial primary production at a given time of year. So they're themselves kind of hotbeds of biological activity. And there's also some evidence, for example, that they can influence vertical nutrient movements throughout the water column as well. So they're ecologically important phenomenon for a variety of different reasons. And because of this, I think there was a lot of interest in sort of understanding DCMs at a broader scale and trying to understand them across lakes. And so this project really came out of the GLEON 16, the Quebec meeting. This was a subgroup of the TTG. And actually, I was able to go back through and we had pictures from all of our brainstorming notes and things like that from that meeting. And I'd also like to take this opportunity to thank my co-authors and acknowledge my co-authors on this project as well. There was a lot of them that were, I think it was a really, truly collaborative effort and a really helpful group of people. So we know a fair bit about DCM formation. So these we've known about DCMs for a long time. They're not new by any stretch of the imagination, not a new phenomenon or a new observation. And we understand that we see these in stratified water columns where essentially we have opposing gradients in light and nutrients. So you have lots of light on the surface that decreases with depth. And when the water column is stratified, we typically see higher nutrient concentrations deep in the water column and lower nutrient concentrations in the surface. And so that situation essentially sets up, because of these two opposing gradients in lights and nutrients, we can get DCM formation because of a couple different things. You can have high in situ growth at that depth where organisms can really maximize light from above and nutrients from below. Modal organisms may actually swim to those favorable habitats. They may actually move to those locations in the water column. And then also, because the water column is stratified and we have density gradients, non-modal organisms may actually sink or rise to their depth of neutral buoyancy, and then they'll aggregate at a specific depth. It tends to be around the thermocline, and so then again we can get these deep peaks forming for that reason as well. And so a lot of what we know about DCM formation has come from single lake studies, or studies where we've really looked, maybe at a handful of lakes at most. But these studies have identified light and thermal stratification as being really important drivers of the formation of these phenomena and also the depths at which they form. But as a group in this GLEON 16 sort of brainstorming session, we realized that we don't really have a very good understanding of how that scales across lakes. So if light and temperature are important, they also strongly co-vary in the water column, so you can't really tease them apart very well. And what does that look like across a broad variety of lake types, like lake size or lakes of different trophic state? And we really realized that we didn't really have a great understanding of that. And so that's really kind of what we set out to answer with this project. We formalize this basically into two different questions. What is the relative importance of light availability versus the depth and structure of thermal stratification in predicting DCM structure? And then again, what does that look like across the broad gradient of lake types? Is it light always more important, or does it change depending on what kind of system we're looking in? Okay, there we go. And so again, we took advantage of the GLEON network. And I think GLEON is really uniquely poised, as has been discussed in a lot of the other talks today, to answer questions like this at this scale, at this really broad scale, and think about lakes as populations rather than just looking at it at a single individual lake. And so we solicited to the network for data on chlorophyll fluorescence profiles, temperature profiles, and light information. And we ended up with a data set of 100 unique lakes. This represents data from 8 countries, a little northern as usual, a little northern hemisphere heavy, but this represents about 13 different ecological regions. So we have a pretty broad variety of lakes in this data set. The first thing that we had to do, which was actually a challenge I had not anticipated, was defining what is a DC, like when does a vertical profile have a DCM? I think we all kind of look at vertical profiles and just sort of have this gestalt like, oh, that's a DCM, or oh, that's not. But when you have 100 profiles or more, how do you define that in a way that's maybe a little bit more objective? So that was actually our first challenge, which we overcame, and I won't get into the details of exactly how we defined it. But from there, we were then able to look at 2 different aspects of DCM structure. DCM depth, which has been relatively well studied, but across this broad gradient of lakes, there's other features of the DCM that may be ecologically important, and so we wanted to understand that at this scale in a little bit more detail as well. And the one thing we focused in on was the thickness of this layer. So you can have, the depth of this maximum, but it's typically a gradient. And so how thick that layer is may have ecological relevance to a number of different processes as well. And as with defining whether or not something has a structure, defining the thickness when you have lakes that are, 8 meters deep versus lakes that are 70 meters deep is proved to be, again, a little bit more of a challenge. And what we ended up doing was using a curve fitting procedure to fit curves to these chlorophyll profiles, and then using parameters from that curve to define the thickness of our DCM. And so once we had those 2 variables, the thickness and the depth of the DCM, we used a random forest analysis to try to tease out which variables are most important in predicting these two features of DCM structure. And again, I'm not going to get into the details of the random forest analysis, but I'm happy to answer questions about it or talk to people about it afterwards. But a random forest analysis is a machine learning technique that is very good at giving you variable importance so you can feed it a bunch of different predictors, and it will essentially give you a ranked importance of all of those predictors in predicting the response that you're interested in. And so it's really good with variables that are collinear with one another, which is really important in this case because light and temperature are very collinear. And it's also pretty good. You don't have to assume anything about linearity of relationships between predictors and responses as well a priori. So it has a couple of really nice features with regard to that. And so we were trying, again, because we're interested in light and light associated variables versus thermal stratification. We looked at some of the predictors we fed into this model were the depth of 1% par, DOC concentration, metrics related to lake size, including surface area and depth, and then also a handful of metrics related to the depth and strength of thermal stratification. And what we found was that DCM depth is by far predicted by the 1% par depth. And so as lakes get clearer, if they have a DCM, their DCMs get deeper, which is not a huge surprise, but I don't think anyone had ever really shown that at this scale. And so another interesting thing that came out of this, because we can look at this population of lakes, is that while DCMs are typically considered to be associated with the 1% par depth, there's actually a pretty big range in the percent par available at the depth of the DCMs across all of these lakes. It goes from about below 1% up to about 25% or so. So again, there's differences across lakes in how much light is available. And I think that might reflect some species differences in sort of where they're balancing light availability versus nutrient availability. And also it can reflect change differences in the actual amount of light versus percent par available at those depths. And again, because we know that DCMs are typically associated with, can often be associated with the thermal climate, what we see is in the vast majority of lakes, DCMs are located below the thermocline, and in all of the lakes, they were located below the top of the metalimnion. So you really don't see these phenomenon unless you're at least within the metalimnion or well below it in some instances. Another nice feature of the random forest analyses is that we can actually use them to look at the sensitivity of our response to a given variable, all other things held constant, all other potential interactions among predictors held constant, which is again a nice feature when you have these environmental variables that tend to covary pretty strongly. And so this figure here is what's called a partial dependency plot. And so you can think about this as the sensitivity of DCM depth of our response to changes in 1% par depth, for example. And so anywhere where you have sort of a flat line that indicates that changes in 1% par doesn't really do much to our response. And what we can see with this is that there seems to be some nonlinearity here, that in lakes where the 1% par depth is between about 10 and 30 meters deep, which are pretty clear lakes, the DCM is pretty sensitive to changes, more sensitive to changes in light environment than on either end of that as well. So again, light's really important, but it's the sensitivity of the DCM depth to light varies across lake type, which I think is interesting and something we wouldn't have known had we not been able to look at this broader population of lakes. When we look at DCM thickness, actually we see a very different story. So our random forest, the metric that came out is, again, hands down the strongest predictor of DCM thickness was actually surface area. So the size of the lake, the surface area of the lake, with larger lakes tending to have thicker DCMs. And I think that this really probably reflects the influence of lake size on several different aspects of mixing and of thermal stratification in lakes. So large lakes tend to be less wind sheltered. They tend to be much more subject to wind mixing, and oftentimes more weakly stratified than smaller lakes as a result. And so when you have this weaker stratification, that sets up the possibility that over that depth, from that depth where light and nutrients are optimized, you can have more movement of organisms, either or because of they're aggregating there because of neutral buoyancy or because of other mixing processes happening. And again, we can use the partial dependencies to explore this in a little bit more detail. And again, you can see here that our response, so DCM thickness in this case, in these really, really small lakes isn't super sensitive to surface area. So it's probably not as sensitive, the thickness isn't as sensitive to some of these mixing processes, which makes sense because they're not as important in these small lakes. Versus when you get to about 1 kilometer squared or so, the response becomes much more sensitive to surface area in that region. And that's actually a really interesting cutoff, because that same cutoff at about 1 kilometer squared has been identified in other studies that have looked at the importance of physical mixing processes as controlled by lake size. So again, when you get into these larger lakes, these physical processes tend to dominate and tend to be more important. And we see that in this instance as well, which I think is pretty cool. So some of the main takeaways, again, DCM depth is largely controlled by light, but it doesn't really seem to be linear across lakes. Whereas the thickness of these structures is much more controlled by lake size, and likely because of changes in the thermal structure. And again, there's differences across lakes in how important these mixing processes may be to some of these DCM structures. And I think actually one of the really cool things that comes out of this study, which is kind of an interesting aside really, is that DCMs are actually common across a lot of different lake types. So I think we typically think of them as being associated with these really clear oligotrophic systems. But if you look at our data set, there's a lot of mesotrophic and even a handful of what we would consider eutrophic lakes in this data set that have DCMs. And so I think that's a really important takeaway, again, from being able to look at this large population of lakes, that when you're thinking about productivity and you're thinking about vertical gradients in your lake, you may have to consider deep production in situations where maybe we wouldn't typically think about that, at least at certain times of the year. And so as I mentioned, this project was actually, we've published this recently in LNO. And again, I'd like to acknowledge my co-authors here. And I think with that, I will skip the last part and ask if there's any questions.
Thank you very much, Taylor. Still a very interesting story. Of course, I had read the paper, but I really like the whole DCN story. And yes, if there are any questions, we can use a few minutes to discuss. If there's nobody else, I would have a question, Taylor. So in the National Lake Assessment, right?
Yep.
The 1,000 lakes that the EPA samples, if I'm correct, they sample only the top 2 meters. So clearly, in the majority of cases where there is a DCM, they would miss that. Would you have an understanding now? So if you take 1,000 lakes, say half of them are deep, how many lakes do we have a serious underestimation of chlorophyll and productivity among the 500 lakes in the NLA?
That's actually a really good question, and one that I don't think we're quite poised to answer with this data set, just because of the way that we solicited for data. We asked people to send us data where they thought they had DCMs. So I think that if we had solicited more broadly, just if you have chlorophyll profiles, we may have been able to get at that a little bit more. So I don't think I can give you a number, but I can tell you that based on what I've seen with this data set and with some more recent data sets that I'm working on from lakes in the Adirondacks, that I think actually DCMs are pretty common, especially in the middle of the summer in a lot of different lakes, even lakes where you would think, oh no, those are way too dark to have DCMs. I think they're actually pretty common. And it would be an interesting follow-up project to this, is to really get an estimate of that, because I think you're right. I think we are missing a lot of deep production in some of these systems, particularly peak summer.
I was going to ask if you would be interested in such a follow-up, because I would be. And for a second reason, clearly we understand that if lakes are turbid, that may be prohibitive for a DCM to develop, but we have at least one example now of a deep lake in Switzerland that has become too transparent for a DCM to develop, because now it sits so deep in the lake that it gets to a region where there's a lot of shear stress really at the bottom of the thermocline, and then there's too much displacement for the DCM to remain intact. So it's a lake where the DCM has moved by 7 or 10 meters, I think, over the last 10 years because of the lake getting more transparent. I'd be really interested to see if there's more cases like that. But
yeah, I mean, I think that would be a really great follow-up project. So definitely be interested in. I mean, I think it's an important question, especially when we're looking at these large population of lakes. I think it's important to understand what we're missing if we're only sampling.
Okay, so thanks again, Taylor. I guess, yeah. So I can see yesterday.
Can you see it now?
Yes, you're fine now. So go ahead.
Yeah, we good?
Yeah, we're good.
Oh, great. Okay. Okay, so I hope you can hear me. So my name's Iestyn Woolway. I'm a postdoc at the University of Reading in the UK. And my research involves investigating climate-related changes in lake surface water temperature, specifically looking at using satellite data